Bismillahirrahmanirrahim. Assalamu alaikum warahmatullahi wabarakatuh. This is Abdurrahman Kahi and you are watching Animals Production Practices. In our today lecture we are going to talk on the glycogenesis and the glycogenolysis. This is the most important lecture regarding the nutrition of the animal and the metabolism of occurring within the animal. And the story begins with the term glycogen. What is glycogen? It is the glucose polymer. Glucose attached by the alpha 1 to 4 linkage or alpha 1 to 6 linkage to form glycogen. Glucose attach by these linkages to form glycogen. However, if the glucose joined alpha 1 to 4 linkage, then the straight chain is going to form. However, if the glucose attached with alpha 1 to 4 linkage, and the branch chain are going to be formed. And glycogen synthesis mainly occur in the cytoplasm of the liver and the synthesis and the skeletal muscles. The muscle glycogen provides readily available source of glucose 1-phosphate for glycolysis within muscle itself. However, liver glycogen functions as a reserve to maintain blood glucose concentration in fasting. Glycogenesis. Enough glucose and energy triggers the synthesis of the glycogen, which is known as glycogenesis. A family of facilitative glucose transporter is involved in the regulating tissue-specific glucose uptake and metabolism in the liver, skeletal muscle, and adipose tissue. The glucose transporter isoform GLUT2 is expressed in the liver, intestine, kidney, and pancreatic islets of beta cells, as well as in the central nervous system. The last mentioned glucose transporter GLUT4 is known as major isoform in muscular and adipose tissues and only shows minor expression levels in the liver as well. What is glycogenesis? Glucose is transported into the liver cells by a specific glucose transporter whereby it is immediately phosphorylated. Hence, most of the glucose in a cell is in the form of glucose 6-phosphate. Conversion of glucose 6-phosphate to glucose 1-phosphate is the very first step in this regard, and the enzyme involved in conversion of glucose 6-phosphate to glucose 1-phosphate is phosphoglucomutase. Remember, this is the reversible reaction in which the glucose 1-phosphate again may be converted into glucose 6-phosphate. It is a reversible reaction allows glucose 1-phosphate conversion to glucose 6-phosphate in glycogenolysis. Glycogenolysis is the reverse of the glycogenesis. Synthesis of uridine diphosphoglucose UDP. Enzyme involved in this regard is UDP-glucose phosphorylase, pyrophosphorylase. Glucose 1-phosphate that was formed in the earlier slides is now going to be converted into UDP, uridine diphosphoglucose, in the presence of UDP-glucose phosphorylase, pyrophosphorylase. What is the importance of UDP? It is the intermediate of glycogenesis required for the production of vitamin C essential for the synthesis of lactose from galactose. This one glucose can be accepted by a specific protein that was glycogenin. To form alpha 1,4 linkage. Glycogen synthase can catalyze the synthesis of a linear unbranched molecule with alpha 1,4 glycosidic linkage. Glycogen is a branched tree-like structure. The formation of branches is brought about by the action of a branching enzyme, namely glycosyl 4,6 transferase. This enzyme transfers a small fragment of 5 to 8 glucose residues from the non-reducing end of the glycogen chain to another glucose residue where it is linked by a alpha 1,6 bond. This leads to the formation of a new non-reducing end besides the existing one. Glycogen is further elongated and branched by the enzyme glycogen synthase and glycosyl 4,6 transferase. Glucose, glucose 6 phosphate, glucose 1 phosphate and glucose is going to be separated from the UDP. And then enzymes involved are also mentioned here: glycokinase, phosphoglucomatase, UDP-glucose pyrophosphorylase. Now the separated UDP is going to be formed or act as a primer to form the long chains in the presence of the enzyme glycogen synthase. Glucose stored in polymeric form as glycogen is mostly in the form is present in the liver and the skeletal muscles. Enough glucose and energy trigger the synthesis of the glycogen, which is known as glycogenesis. However, the glucose can rapidly be delivered to bloodstream when needed upon the degradation of glycogen. This phenomena is known as glycogenolysis. Glycogenolysis, degradation of the glycogen, release of glucose 1 phosphate, and the enzyme involved is glycogen phosphorylase. These are the non-reducing ends. Glucose 1 phosphate always act as a non-reducing end. However, 1,4 glycosidic link is cleaved by phosphorylases with retention of the energy potential in the phosphate ester of glucose 1 phosphate. Stops at 4th glucose from a 1,6 branch point. Contrast to enzymes acting on the starch and glycogen in the gut, which yield sugars, not sugar phosphate, as product. Activated by phosphorylation, regulated by glucagon and epinephrine. Metabolic regulation by glycogen levels. Glycogen reserves are the most immediately available large source of metabolic energy. Its storage and utilization is under dietary and hormonal control. Primary responsible hormones are epinephrine, glucagon, and the insulin. Primary enzymes targets in glycogen metabolism are glycogen phosphorylase and glycogen synthase. Hormones are epinephrine or adrenaline, glucagon, insulin, and the enzymes are glycogen phosphorylase and glycogen synthase. What is phosphorylation? Making the inactive phosphorylase active conversion of the inactive phosphorylase into active phosphorylase is known as phosphorylation, and then that activated phosphorylase is going to be converted into glycogen breakdown. Glycogen phosphorylase is activated upon phosphorylation by the phosphorylase kinase. Phosphorylase kinase is activated upon phosphorylation by protein kinase. This is the phosphorylase kinase which is in inactive form. It is going to be converted into phosphorylase kinase by the protein kinase pKa, and then inactive glycogen phosphorylase is going to be converted into glycogen phosphorylase which is active, and this one was done by the phosphorylase kinase. Glycogen phosphorylase is activated upon phosphorylation by phosphorylase kinase, and then phosphorylase kinase is activated upon phosphorylation by protein kinase. Protein kinase is activated by cyclic AMP, which is produced by a G protein in response to epinephrine or glucagon. AMP is produced by the G protein in response to glucagon or the epinephrine, and this AMP is going to be act as to activate the pKa, phosphorylase kinase. If you clearly attentively look at this picture, you can see there are two sections in this picture, this one and this one. Glucagon, which is hepatocyte condition, and the epinephrine, which is myocyte condition. In case of glucagon, the glucose 1-phosphate is going to be converted into glucose, and then glucose is going to be shifted or transferred to the blood. However, in case of epinephrine, the glucose 1-phosphate is going to converted or go to glycolysis, which is going to be stored in the muscles of the body. So there are two conditions or two fates of the glucose phosphate in two conditions. In case of glucagon, the glucose is going to shifted or transferred to blood, and in case of glucose 1-phosphate is glycolysis is going to be converted or shifted to muscle contraction. That's all about today's lecture. Thank you very much. If you are having any question, you may ask in the comments.